Have you ever actually stopped to think about the hidden biology literally surrounding us every single day? We're not just talking about those towering trees or the colorful flowers you see in the park. Oh no, there is a whole invisible world operating right under our noses. So today we are going on a proper scientific mission to uncover it. We're going to dive into the secret and honestly super fascinating world of fungi. And the best part, we're doing it by stepping right into the shoes of a student scientist. Let's get into this explainer. To kick things off, I've got a central question for you. Think about this. How exactly do living things reproduce without seeds? It is such a fantastic biological puzzle, right? We are so used to the typical cycle of nature. You know the drill. A flower blooms, it produces a seed, and then bam, a new plant eventually grows from that seed. But what happens when an organism just doesn't have flowers? How does life find a way to multiply and spread all across the globe without that familiar seed mechanism? Keep that question in the back of your mind, because solving this biological mystery, that's the entire core of our exploration today.
Okay, so look at this actual Science IE homework assignment poster from Montfort College. It brilliantly lays out exactly how we're going to crack this case. And it's not just some dry reading assignment either. It is a full-on, hands-on, adventurous mission. Starting the week of June 22nd to the 26th, 2026, students are literally tasked with bringing the laboratory straight into their own homes. The mission here? Prepare mushrooms for scientific study. I just absolutely love this approach. It takes everyday, normal household items and turns them into profound tools for learning and discovery. So, our scientific objective is crystal clear. We need to observe and record the hidden characteristics of mushrooms and spores. We are definitely not just chopping up veggies for dinner here. We're executing a highly specific scientific protocol to reveal biological structures that are, get this, normally completely invisible to the naked eye. We are actively hunting for the very mechanism of fungal life itself. But hey, before we start hacking away, we really have to establish the #1 rule of fieldwork, and that is safety. The protocol strictly mandates that we choose only safe and edible specimens, things you can easily just grab at the local market. We're talking oyster mushrooms, straw mushrooms, wood ear mushrooms, or shiitake mushrooms. This is absolutely vital. Out in the wild, some mushrooms can be incredibly toxic. So by controlling our variables and using known, totally safe, edible mushrooms, we make sure our experiment is super educational and completely hazard-free. All right, so here is our precise 6-step preparation method. Ready? First, we take those safe mushrooms, wash them clean, and slice them paper thin. You want to really increase that surface area. Then, leave them out to dry in the sun completely. That draws out all the moisture and actually releases the very things we're hunting for. Finally, they get sealed up tight in a clean, dry container, and you label them. It's a surprisingly elegant process, actually, but it does require the patience and discipline of a true scientist. Now, notice the intense focus on that final step, the label. Seriously, an experiment without properly recorded data? That's really just messing around in the kitchen. Proper scientific labeling is literally what transforms a simple jar of dried fungi into a highly valuable scientific specimen. Just like the curriculum outlines, we've got to record the mushroom's name, the exact date it was collected, and the specific place it was found. This kind of meticulous record-keeping is the absolute bedrock of the scientific method. It ensures our observations are actually accurate. Okay, so after all this washing, slicing, drying, and meticulous labeling, what's the ultimate prize here? What are we actually hoping to see when we look closely at these prepared specimens? Well, the answer is spores. Spores are this truly remarkable reproduction method used by organisms that don't have flowers, and it allows them to multiply entirely without seeds. When we dry out those mushroom slices in the sun, millions, literally millions of these tiny, microscopic particles are released. They act kind of like the fungal equivalent of a seed, but mechanically and biologically, they are a completely different survival strategy. Which brings us right back to the question we asked at the very beginning. So on one side of this huge biological divide, we've got organisms that reproduce by spores. And remember, these organisms have no flowers, ever. On the flip side, we have organisms that reproduce by seeds, which are fundamentally the product of flowers. It is such a stark, fascinating contrast in the natural world. Most of the plants we actively grow rely on that classic flower-to-seed pipeline. But fungi, along with several other ancient organisms, they rely entirely on the incredible, microscopic power of the spore to carry on their lineage. And we can actually see this divide all around us. For example, under spore producers, it's not just mushrooms. We've also got things like the bird's nest fern and moss, both thriving without a single flower. Now, contrast that with the seed producers, the sunflower, the rose, or that big tamarind tree.
those are your classic flowering plants. By executing our little mushroom preparation experiment, we're getting a first-hand, highly tangible look at a reproductive strategy that is completely separate from the colorful, blooming plants we usually notice in our gardens. So we have journeyed from a simple piece of school homework to uncovering, well, one of the most fundamental and ancient mechanisms of life on Earth. To wrap up our explainer today, I want to leave you with this fantastic, inspiring quote straight from the Montfort Mission Briefing. It says, "Science is everywhere. Let's explore the secrets of mushrooms together." I just think it's such a powerful reminder. You don't need some multi-million-dollar laboratory to do real, meaningful science. Sometimes, literally, all it takes is a keen eye, a structured method, and a common edible mushroom to unlock an entirely new biological world. So my question for you as we close out is this. What other invisible mysteries do you think are waiting to be discovered right in your own backyard or even your kitchen? Keep observing everyone and keep learning.